Myra, you're here in this batch for the backup classes.
Yes, ma'am.
So what happened? Why weren't you joining the class? Walaikum assalam. Fahad.
Ma'am, actually, I was having exams, so I
Okay.
missed the classes.
Okay. So Hania was also in the same school, no? Sumaira?
Yes, ma'am.
Okay. So how was your exam, both of you? Physics exam?
No, it was not that good.
Okay. So submit your, just share your question paper with me, Sumaira once. Okay, with your prior question paper, I'll have some idea about what type of questions have been asked in your school. Otherwise, any updates about Hania?
Like what, ma'am?
In this, like for exam only. What about her exam?
Ma'am, actually, she's not in my class. She's in the other section.
Okay. I'll talk to her then. I'll contact her. No, not today. Fahad, no test today. Okay, class, let's start with this topic, optical instruments. So, see class, before we start with the actual optical instruments, no, let us just quickly see some basics about optical instruments, then we can proceed with this. So, optical instruments are basically, these are devices that make use of mirrors are also present, lenses are also present. Fine, prism is also present. Prism we'll do, prism is still left to be then done in this batch. So they are basically used to extend the human eye vision. Now, extending human eye vision means what? When I say extending human eye vision, that simply shows if I'm able to see at this much distance, let's say, after beyond this distance, I'm not able to see anything clearly. Let's assume it. So, what will happen if a tiny micro object is present, microparticle is present? How will I see it? Or let's say something that is kept far, very far away, not even this much distance, a distance that is far more greater. Then what we'll do? So to enhance the vision, the range of the vision, that is, let's not quickly just take onto this vision only, to expand it. Like let's say about the celestial objects that are present. Are we not able to see celestial objects through telescopes? So telescopes are what? These are basically optical instruments. So optical instruments are devices that makes use of mirrors, lens, prism, all these just to extend the range of human vision. Class, you have two instruments. You have basically two types of instruments. One is your microscope, which we'll study, microscope. Other one is telescope. Microscope are also of two types. Compound microscopes are there. Then you have simple microscopes, both of it which we'll see. Now see, human eye is not there included in your course now. Human eye has been deleted. But just the basics of human eye is that your eye lens, which you are able to, by which you are able to see objects clearly, those are actually convex lens we have studied in detail. So those are convex lens. So lots of things are present when you draw the actual structure of eye, which I'm not drawing it. Cornea is there, scleroid layer is there, choroid membrane is there, so many layers of eye, 3 layers I think of eyes are there. And then this is the lens that is present through which refraction occurs, between which you have aqueous humor. This chamber is known as aqueous chamber. Then after aqueous chamber, you have the retina behind where the image ultimately forms. So this is known as vitreous chamber, which is filled with vitreous humor. Humor is basically used to denote fluid, like blood, we say it's humor, like this. Then at a certain point where no cones are present, you call it as blind spot. Below it, you have optic nerve that carries whatever impulses are made, because in the retina, inverted image is formed. So obviously, inverted image, if we see inverted image, we'll not be able to grasp anything. So it takes it to the cerebral cortex, wherever the visual areas, motor areas are present in your brain, and so the image is interpreted. Like this is the working of eye. So in this entire pathway, in any point you have any abnormality, let's say lens is not able to converge properly, light rays are not able to enter it, retina is not able to transmit the image to the optic nerve, like this, anything that happens, that this causes abnormalities. So hypermetropia, myopia, all these diseases are present. Right now, this is just enough in the introduction of eye. Now you don't need to know anything about eye. Human eye, I'm again saying it is deleted. If you'll open your NCRT, in NCRT books, it is mentioned. It is still given in NCRT books. So do not study that part. It won't be ever asked. Near point of eye, yes, these 2 points are very important. Near point and the far point. We'll be dividing the whole topic now of optical instruments, and each and every optical instruments based on near point, then far point. Like for example, let's say simple microscope. So first we'll see the case of near point, then we'll see the case of far point. This will be valid for all the optical instruments. Now, near point is the closest distance from the object, right? A closest distance of the object from the eye. Let's say this much distance. So at this particular distance, I am able to see my hand clearly. Fine. But when I am shifting it till this distance, let's assume I'm not able to see it very clearly the way I was able to see it. Or hand, or even it's better if you take pen. At a certain distance, you are able to see pen clearly, right? Then a distance comes when it start becoming two, you see, then it becomes blur, all these things. So the point where you are able to see it clearly, that is known as near point of the eye. So this is also known as least distance of distinct vision. Least distance of distinct vision. And this is 25 centimeter. I'll be using a capital D to denote it. So please remember this value, 25 centimeters. It won't be mentioned anywhere. In some questions, it can be present, it can be mentioned, but this value you have to remember. 25 centimeter is the least distance of distinct vision. Means at this particular distance only you are able to see any object clearly. Now opposite to it, far point. How much far your eyes can see? 5 meters, 10 meters, 18 meters, 100 meters, 1,000 kilometers. How much your eye can see far? Obviously, it is infinity, this distance, class. See, are you not able to see the moon? Are you not able to see the sun? Celestial objects are visible to you as well, right? I'm not saying with exactly same size. Definitely, they look smaller when you'll see it from here. But they are visible and they are directly visible. You just look up in the sky, you are able to see the moon clearly at night, right? The daytime, you're able to see the sun. So it means that distance is infinity. Till infinity, your eyes can see in a straight line, provided that it is a straight line. You cannot see objects that are kept behind it. So far distance is infinity. Now we will start with simple microscope. C class, just telling you again, microscope is divided into 2 parts. Simple microscope is there that uses 1 lens. Compound microscope is there that uses 2 lenses. Now for each one, we'll see both the case. If the final image is formed at least distance of distinct vision, and when the final image is formed at infinity. It means when the final image of the whole optical instrument is formed at near point and one at far point. Got it? Same thing for compound microscope as well. Near point, when the image is formed at near point, and when the image is formed at far point. This we'll see. I'll just note down these introductory points so that I can start with final image formed at least distance of distinct vision for simple microscope. One case now, if you do it properly, least distance 1. Infinity case is very easy. Infinity case becomes very easy then. And also class, optical instruments, you don't need to use sign convention. That I'll tell you. Let us just write it down. Let us just start with microscope. Samira, I hope this part is not done with you.
No, ma'am.
Also, class, please update me about your pre-boards. Samya, in the group, I have shared the link for ray optics. Just check it once. I have shared the link in the group. On both the groups, I have sent it, boys and girls both. Just check it once. I have written also class notes, something like this. Drive, OneDrive is there. I was saying, class, please tell me about your pre-boards whenever you are having 5th Feb. Okay, Rifat. Pre-boards are after my board practical exams. No, Madiha, I mean, I want to ask, acha, okay, board practical exam. Okay. So board practical exams are when? At least month, some idea? Because see, after this, ray optics is almost completed. Prism is the next topic that is left. So, and the rest of the topics will go on fastly. So I just wanted to know about your months of yes, model exams. The exam that you have before the board exams. First 2 weeks of Jan. Okay. First 2 weeks. So after first 2 weeks. So mid Jan. And Sahal first Feb. So mostly Jan and Feb, no? Okay, Priya. If you all will get the date sheet also, no, so just let me know. Okay, so probably, yeah. Then it will be around after 15th only. Because if it is started, if it will be in the middle of it, so 15th it will take. 15th it will. So just update me, keep on updating me about your pre-board exams because I have to plan the revision according to it only.
Ma'am, should we draw the diagram before the near point?
No, you can leave that. C class, just understand the first one at least with patience. Wherever you are having difficulties, any doubts, get it clarified. Okay, final image is formed at least distance of distinct vision. It means final image is formed. We are considering the case when we are able to see things clearly. So final image is formed at least distance of distinct vision, and we are using microscope. Microscopes, I think all of you must have used once, or I think in 9th or 10th standard also. Which one, Fahad? Just let me know. I'll scroll up. This initial, don't draw this diagram of I, no need to draw. Just write these points. Students who must be having biology must have used microscopes, and students who don't have bio right now but have had biology in class till class 10th. So I think in 9th or 10th also we use microscopes. So those same microscopes we are going to discuss about. Fahad, just let me know once you've written. I'll start. C class, if the microscope is not there, let's say this is the object AB, so it will be subtending some amount of angle. At the human eye, let's assume human eyes are here. So it will be subtending some angle. Let's say this angle is alpha. Let's name this point as something. Oh, let's write it as O. Now I am drawing the diagrams for the actual array. So convex lens is there. Now see, object is kept. This was the case. This I have drawn it without the lens. Now lens has been inserted. So, okay, now the ray diagrams will begin. It will require more space. I'll draw the object near only. Let me draw it near to the lens. So one ray has passed through the optical center. So as I have told you, when it will pass through the optical center, it will retrace its path. And the other one will be passing through the convex lens, and this will get refracted. Here we'll be able to see it. This is the placement of human eye. Now see, how will you see it? No lines are intersecting, so just produce it. These lines are there, now just extend it. These lines also that are present, just extend it, produce it, and see where are they meeting. So this will form the image. A dash, B dash, this will be the image. Fine. So this is how you draw the ray diagram for it. Now see, this separation is what? This is the least distance of distinct vision. Why? Because final image is formed at least distance of distinct vision. So V, that is the image distance from the image, this image distance. This is taken as what? This is directly taken as capital T. This was object distance here. This is object distance over here. Now see the angle that is subtended by the object on the human eye. Now see, now understand. The diagram part I have drawn, now this will become quickly. This will get over quickly. See, object, the angle that the object will subtend at the eye. This I have taken as alpha. And one angle will be there that will be subtended by the image on the human eye. Let's say this angle is beta. Seen, this angle is beta, this angle is alpha. Now class, this is the ray diagram. Ray diagram will be drawn like this only. But one term you have that is known as angular magnification. So this is the ratio of angle subtended by image and object on eye when both are at least distance of distinct vision. So see, here the object is not at least distance of distinct vision. Why? Because we have kept the object, we have adjusted the object so that we could see this clearly. And to see the image clearly, to make the image at least distance of distinct vision, we had to shift this. But here, without the lens, we could see the object clearly. It means this was the least distance of distinct vision. So class, pay attention here. Understand the meaning once. See, you just have to understand the meanings once. Just then after this, you just have to see the modifications. Angle subtended by image on the eye. Divided by, means it is the ratio, no? So ratio of angle subtended by image on eye when image is at least distance of distinct vision. This separation is least distance of distinct vision. Divided by angle subtended by object when it is at least distance of distinct vision. It means alpha. So angular magnification is what? This is different from linear magnification. Angular magnification will become what? Magnification will become beta divided by alpha class. Now see, for the calculation purpose, we can do one thing. We can take tan beta approximately equal to beta and solve this, because something we have to keep in here. Now see, if you look at these triangles, this is tan alpha. So alpha is equal to what? AB divided by OB, right? AB divided by OB, because this is the angle subtended. This is AB divided by OB. This is angle subtended by object. Then you have angle subtended by image, that is beta. So tan beta will be what? Tan beta will be what? Tan beta will simply be A dash B dash by O, right? A dash B dash by O. See here. Now see, we'll just substitute this in the triangles that are formed. So this will almost be equal to tan beta by tan alpha. Reason behind this is class, tan alpha, tan beta, tan theta, any tangent of angle when it is small. Small means when it is less than 15 degrees. If it is small, then it can almost be equated approximately to theta only. So tan theta becomes almost equal to theta. Right. So see, if even if you take, let me write the ratios directly over here for magnification. So magnification will become what? Magnification will become AB by OB multiplied by OB dash by A dash B dash. Right. This is what it will become. Now see class. Here A dash B dash, or here you can see. This can also be written as AB. AB is the perpendicular distance, right? In this triangle, this is AB divided by OB, right? This is also AB divided by OB. See here. So see, yes, one more thing. What all terms are over here? OB dash. If you look here, OB dash. OB dash is what? OB dash is simply the least distance of distinct vision, which is capital D. So capital, and one more thing, class, one more thing. Capital D's value you have to remember, that is 25 centimeter. Fine, this value you have to remember. And also see class, this I know this is held close to the lens. So angle subtended by the lens, it may be the angle that is subtended by the eye. Do keep in mind, because the lens is kept very close to eye. Don't go by this distance. Okay, do not go by this distance. This is not a very huge distance. This is a smaller distance only. Understood? This is a smaller distance only. Right. So here now what will happen? See. Yes, my dear. Sure. Okay. Yes, Fahad. See, that is the definition. See. That's what I was telling you. The ratio of angle subtended by image. See, this image, this is substituting this angle beta. This image, this final image, this is substituting some angle beta on the eye. Fine. This angle that is substituted by it. Whereas the angle that is substituted by the object on eye, this angle, the angle that is A dash where? This is A dash B dash. Final image. This is A dash B dash. This is AB. So A dash B dash is subtending angle beta at O. Fine. AB is the object that is subtending angle alpha at O. Why did we choose this? We have chosen this separately because C class. If I choose and I keep this object here only, there's a difference. I'll not be able to see this final image clearly. Do you notice? I have to adjust. Whenever you're using microscope, you have to keep on adjusting. In one go, you're not able to see the image clearly. And at any position, you're not able to see it clearly. That adjustment of microscope is this only. No doubt, Fahad, which you're asking. This is the adjustment of microscope, basically. So you keep the object in such a position where you see the image clearly. And at what point will you see the image clearly? At what particular point? When it is at least distance of distinct vision, right? So that's why A dash B dash is the final image. A dash B dash, this is the final image that is formed. Understood? This is the final image that is formed. And angular magnification is the ratio of this. Angular magnification is simply, this is the ratio of this. So yes, now I hope this is clear. And definition of angular magnification, let us complete with these steps. Now see what you were asking. Beta by alpha will become the same thing then, angle subtended by image, angle subtended by object. Now, I was saying if you have small angles, tan theta can almost be written as theta. So theta also can also be written as tan theta, no? So it means I have beta, I can write it as tan beta. I have alpha, I can write it as tan alpha. That is what is the difference. So yes, this was beta was what? Angle subtended by the image, right? So see, this becomes what? This was A dash B dash, no? A dash B dash by O B. And tan alpha was OB or what? What was A dash B dash by OB dash? Okay. A dash B dash by OB dash. And tan alpha was AB by OB. So this becomes OB by AB. Now see class. For the derivation purpose, now what do? We choose this particular point over here. Right? This particular point at which the object distance is lying. Because when we draw both of it together, the question that you were asking, if you draw it together, then this will how the ray diagram will look like. See class, this will become alpha, this will become beta. Name this something. Let's say double dash. So that's why I have drawn it separately. This is the same thing that is kept. Understood? This is the same thing that is kept. So A dash, B dash also you can use for tan alpha. If you wish, you can also use A dash, A double dash, B dash. And this A double dash, B dash is equal to AB. Okay, now clear, no? Okay, yes, that's what I was saying. A dash, B dash. No, even if you wish, you can draw both of it together. Same answer will come. See, now magnification will be what? Right, AB into OB by OB dash into AB cancels each other. Magnification. What is the magnification that is there? Magnification will be OB dash by OB. And OB dash is the least distance of distinct vision. So that will be, let's say, minus D. OB is the object distance, so minus U. But see class, we will not consider this as final. Let us use lens formula. Lens formula is what? 1 by F is equal to 1 by V minus 1 by U. Focus of it, keep it as F. Image distance, you know, this is the least distance of distinct vision, right? Object distance we are trying to substitute in here. So if you try to substitute your object distance, this will become 1 by D plus 1 by F, because the negative will become positive. So class C, this is 1 by U, 1 by D plus 1 by F. This is the value of 1 by U, no? Now see, just take the reciprocal of it, or just take D common from here. If you take D common from here, what will you be left with? See, D by U will be there on this side. We or do one thing. D is here, D by 1 by U is there. I can write this magnification as D by 1 by U. This magnification class. This magnification I can write it as D by 1 by U as well. D into 1 by U. D by U I write, or D into 1 by U. Now 1 by U's value I have. This becomes D 1 plus D by F. Now this is the final formula for the magnification when at least distance of distinct vision. One more thing is possible, take D common. So that if you take D common from both the sides, this will get cancelled because it will be common in the denominator. So you will be left with 1 at this side, or just multiply D within the bracket. You will get D by F. This is the formula of magnification. No sign convention. No sign convention. Do not use sign convention. This is the formula of magnification. So since this was the first part, that's why I had taken time. I have repeated it a lot. Still, if you have any doubts, let me know. Copy it. Now this will become a little faster. Note it down, class. See class, if this is the case of infinity, no, then what will the difference be here? So I'll just make the correction here only. If it is case 2, final distance is at infinity. See, object distance, there won't be an effect on object distance. But regarding the image distance, see. This won't be least distance of distinct vision now. See, rays will come. Will pass through the optical center. Now see, when you will produce it, this won't appear to meet anywhere. So the image will be formed at infinity. This will be the difference that will arise. Understood? And now see, if it is formed at infinity, it means the object distance becomes the focus. If your object is kept on focus, then only you are getting the final image at infinity. This will be the difference that will arise here. Tan beta by tan alpha, these derivations will be the same. But C class, one thing, if you notice here, what will be the difference? Instead of the object distance directly here, you will get your answer. See, I'll just show. No need of all these steps. Your answer will be completed till here only. C class. OB dash is what? OB dash. This is still least distance of distinct vision. But OB, that is the object distance. This is what? This is the focus. So magnification becomes D by F. That's it. All right, quickly note it down. See class, let's just do one numerical from this as well. A converging lens of focal length 6.25 centimeters is used as magnifying glasses. Near point of observer is 25 centimeters. So this is given in the question. Focal length is given to you. Near point, capital D, least distance of distinct vision is given to you, and it is asking you distance of object from the lens. See, first part is involving U is missing. V is given as 25 centimeters. Focal length is 6.25 centimeters. So which formula should be applied here? Yes, class. Which formula should be applied over here? Yes, Priyadarshini. Correct. Lens formula will be applied. So let's put in the sign convention. Focal length is behind, so this will be positive, but this will be negative. So 1 by U will become 1 by V minus 1 by F. So 1 by minus 25 minus 1 by 6.25, right? So see, minus 1 by 25 can remain. So 100, then 4, then this can be minus 4 by 25. So this is minus 1 by 5. So 1 by U is minus 1 by 5. U will be what? U will be minus 5 centimeters. Second part is angular magnification. It means first formula. Magnification is 1 plus capital D by F. No sign convention here. Directly write it. 25 by 6.25. This will be the answer. Third part says magnification when final image is at infinity. D by F only. So 25 by 6.25. No need to use sign convention in these. But sign convention has to be used in lens formula. Note it down, then we'll start with compound microscope. Class, please remember, for rest of the things you have to use the sign convention. Just for the magnification of optical instruments, you can omit it. You don't have to use it. F will be given in the question. Yes, yes. If F is not given, then object distance and image distance will be given so that you can calculate F. Okay, Fahad. Yes. So if this is 5, so then this will be 4. Because here 1 is getting added. Written all of you. Let's start with compound microscope then. Any doubts in simple microscope? Now see, in case of compound microscope, though, now see, when do we use compound microscope? We use when we have to magnify the image by 1,000 times approximately. So how do you construct it? One objective lens is there and one eye lens will be there. Object lens will be closer to the object and eye lens will be positioned closer to the eye so that we can view the image. But see, in case of microscopes, compound microscopes, objective lens is smaller, eyepiece is greater. Understand this. Remember this because in telescope you will have a modification on this. So again, both the cases will require case when the final image is formed at least distance of distinct vision. See class, just draw it properly with the help of scale because on the screen it becomes a little difficult to draw. So refracted ray came, it bent. Then the ray will pass. This has to pass actually through the optical center. Okay. So this will pass through the optical center. It is intersecting here. This shouldn't intersect here. If you'll draw it with the help of proper scale, do one thing class. Whenever for ray or diagrams, I'll suggest you draw the rays first. Then mark the lens over it. Like had I drawn the line first, no, then I would have drawn the lens according to it. Because drawing the lens is in your hand. These lines are fixed. These lines are very rigid. So right now, whenever you are drawing it, if you're drawing it fairly, do mark it. Now see here, one intermediate image is formed, right? A dash, B dash, this is formed. All right, just keep in mind, this is formed. And what about the other images? See, this will get refracted by the eyepiece. See class, this is the eyepiece. This is the objective lens. So what all will be the distances? If the image is formed here, this becomes focus. A dash, B dash I am writing, but this becomes the focus. And this is the image distance of the objective lens. This part is the image distance. Eyepiece lens are big. Yes. Every diagram. Yes. Because in telescopes it will be reversed, but in every diagram. So this is the image distance for the objective lens. See class, just ignore eyepiece. Till here, if you look, this is the normal lens, no, that we have studied in lens maker formula also. This much of diagram we had drawn. Same thing. Forget that this part is here, which is extending. So the same thing, this will become what? This will be object distance, U0, like this, the way you have been using. Somewhere focus will also be there. Let's say if not, focus is also there somewhere, right? But see, now since this eyepiece is present, we have to produce it. So if you produce this one, it will meet somewhere. Let's say this point it is meeting. So see here you are getting a larger image. Let's say this is A double dash by B double dash. So this is the final image that is formed. And this is formed where? Least distance of distinct vision. So this is least distance of distinct vision. Got it? And again, angle subtended by object on the human eye. That is alpha. Understood? If I draw, I haven't drawn it over here. Let me draw it in this diagram only. See class, the angle that is subtended by the object, this will remain fixed, no? On the eye, that is alpha. This will remain fixed because this will always be without the telescope. Microscope. So this is alpha. This is least distance of distinct vision for it. And this is least distance of distinct vision for it where the final image is formed. Understood? So focus will be there. Focus of eyepiece will also be there like this. This is the ray diagram. So this is, now see how final image has been formed. Because this intermediate image in between acted as an object. Right, virtual object we have seen. In the last class, remember we did a question also. Question to combination of lens. So same thing. So it means this is the object distance for the eyepiece. This is the ray diagram for it. Now coming to the derivation. Same formula we'll apply, same thing. Definition will be the same here. So angular magnification will be what? Angular magnification, if you will draw, this will be exactly the same. That is angle subtended by the image on eye when at least distance of distinct vision and object on eye when at least distance of distinct vision. So this becomes tan beta by tan alpha. So magnification will be what? See, tan beta, this is formed by image, that is A double dash B double dash, height of image by the object distance that is covered, that is UE, right? Into tan alpha. Tan alpha will be the object, that is AB. AB will be in the denominator, no, this time. This will be AB and by least distance of distinct vision. Right, AB by least distance of distinct vision. Instead of writing this, I'll write this together. A double dash B double dash by AB into D by UE. See, A double dash B double dash by AB. This is what? This is the normal for an objective lens. This is the normal magnification. So do you notice the magnification is actually magnification of object distance? Object lens into magnification of eye lens. Into magnification of eye lens. This is how you write it. And magnification of objective lens, just take it as a simple normal convex lens you have been studying. What did we see the magnification of it? Remember, image distance by the object distance. Same thing here it will be. And just now we had seen the formula for the eyepiece, that is the simple microscope. What was it for least distance of distinct vision? 1 by D by F. Now focal length will be what? Focal length will be for this one. So magnification's ultimate formula for compound microscope becomes what? Combine both of this, V naught by U naught, 1 plus D by Fe. This is just the formula. No sign convention. I am again repeating. See, one more thing. Sometimes when the image and when the lenses are kept very close to each other, L becomes the total length of tube, which we say. Sometimes object distance is also replaced by L, that is length of tube. This is also replaced by length of tube. So U naught is replaced by the focus of the objective. When this is which case? Till here your answer is solved. This is, that is L by F naught, 1 plus D by Fe. This is a special case. When this case is valid, when object is placed close to focus. Object is close to focus. If by chance length of tube is given, length of tube, basically, let me tell you in the diagram. Length of tube means this separation between both the lens. So somehow length of tube is given, then you can use this formula. Otherwise, the above formula is sufficient. All right, quickly copy it down. See class, if this is the case, the second case, no, which I was telling you, the case of infinity. Now see the difference that will come here. See, rest of these part will remain the same. Final image, if the final image is formed at infinity, is formed at infinity. Second case. Then see, like this you will have the rays. They'll not meet. The image will be formed at infinity. Now what will be the difference? Object distance, this will be equal to the focal length of eyepiece. Same thing, the way we have repeated in case of compound microscope. So what will be the difference in the angular magnification? In this magnification, for this part, for the objective part which we have, this objective part, for the objective part, there won't be any change. But for this part, for final image, we have this, right? For the final image, we have just d by fe. So here also, you will just be having is equal to v naught by u naught into d by f. Just this will get up. Rest of it will be as it is. All right. Note this down quickly as the second part and text me done when you have completed. All those who have completed, you all can leave. One numerical is left from this topic. We'll complete it in the next class on Thursday.
Thank you, ma'am.
Thank you. Well,
Allah Hafiz, ma'am.